next talk is Michael Weinstein from the Cone Lab and the QCB Laboratory.
Okay, so today I'm going to talk about a program called dsNickFury. It's for designing CRISPR targeting. It's essentially a search tool for looking for potential off-targets for CRISPR. So I know this is a bit of a mixed group, and some people may not be familiar with CRISPR, so I'm going to give a very brief introduction to what is CRISPR. First, I'll say it's one of the big breakthroughs in molecular biology and gene engineering. Science magazine named it as one of the big breakthroughs of 2015. Here it is on the cover of Nature, and this cover of Nature also gives a nice diagram of the system that shows that can be used to show a little bit of its function. This sort of translucent blob is a protein portion known as Cas9. This piece of RNA here that's loaded into the protein is called a guide RNA, and together these make up the CRISPR-Cas9 system. So first you have a guide RNA, and as the Cas9 protein goes along the DNA, the guide RNA looks for matching sequence. The protein itself also looks for a PAM site. This site is invariable. The guide RNA can essentially be altered by genetic engineering and cloning. So anybody trying to use a CRISPR system can just set this. These are typically about 20 base pairs long. The PAM site for pyogenese CRISPR is 3 bases long, although one of the bases can be anything. In addition to that, there's the business end of the CRISPR-Cas9 system, which is these paired nucleases that create single-stranded nicks, but they create single-stranded nicks opposite each other. So what you wind up with is a double-stranded break. And where you have a double-stranded break in the DNA, you can add or lose material, or you can even insert material you've engineered. So CRISPR targeting, in essence, how does it work? First off, CRISPR-Cas9 stands for clustered regularly interspersed short palindromic repeats, and Cas9 is CRISPR-associated protein 9. Neither one of these really describes exactly what it's for. What it really is, it's a bacterial adaptive immune system. The bacteria take on little bits of phage DNA and use them to target phage DNA for degradation in the future. So a target for S. pyogenese CRISPR, which is the system most people are using now, although there are others I'll get into in a moment, can be abstracted to just 20 bases, any 20 bases, plus NGG, which is the fixed PAM site, recognized by the protein itself. Targeting is not perfect. So targeting can actually allow a certain number of mismatches in the guide sequence, typically about 3, but it gets a little bit more complex than that. And CRISPR systems exist in multiple bacterial species, and between different species, their CRISPRs can have some pretty radical differences, especially in PAM sequence. So with those two last things I mentioned, that leads into what the challenge was here. Design a system to efficiently search for potential off-target sites, which are near matches in the guide sequence, for intended CRISPR targets with multiple CRISPR systems. And this was actually designed at the suggestion of a friend who's trying to use CRISPR systems from multiple bacterial species to assess their future utility in human gene therapy. So possible approaches to this. One, I could treat it as an alignment problem. This seemed like the easiest solution at the time, or at least the most computationally simple, given that we already have very good alignment algorithms. It would be pretty flexible, but to run a single search would require setting up something like the Burroughs-Wheeler aligner on a node, and that requires significant system resources. Alternatively, I decided I could try and attack this somewhat de novo and treat it as a big data problem. It would be a little less flexible without a lot of work, but I could ultimately get a lot more efficiency out of it, I figured, and I could run it on computing nodes with a lot less resources, so less memory and fewer CPU cores. I decided in the end to go with a big data approach because I actually thought this would be an interesting experiment to try. So I said I could abstract a CRISPR target into some maximum guide length RNA and some degenerate nucleotide sequence PAM site, so essentially a limited subset of possible nucleotides for the PAM. And I could treat the genome as just a really long string that has several potential CRISPR targets. So this is just a sample of the genome. This is actually pretty much a random sequence. This is about 1/100 millionth the size of the current human genome draft. And so here we see there's PAM sites everywhere there's a GG. On the opposite strand, there's also PAM sites. Here it shows up as a CC, and they go in the opposite direction. And I'm just collecting these from the entire genome. And you can see this is what a typical site would be stored as. And I have to run this again for the entire genome. So I call this process genome indexing, and the minimum amount of information I have to store on every site is what's its sequence and where's it located. I also say some other data, and I save it in a little bit of a different way to make it optimized, but this is essentially the minimum amount of data I need. but is there a way I can speed up the indexing potentially? So it turns out, yes, there is. I can search different chromosomes independently, because finding a target on chromosome 1 and finding a target on chromosome 21 can be done without any information shared between the two. But I can also find potential sites on chunks of chromosomes. So I can chunk out chromosome 1, which is very large in the human genome, and search different parts of it at the same time. This just requires being a little bit more aware of where the borders are between the chunks. And it turns out this can be executed in a very similar manner to a MapReduce job, which is taking a very large set of data, breaking it out into a bunch of independent little jobs, and farming the little independent jobs out to separate computers. So essentially what I'm doing now is, obviously this is more, in real life this is more like 200 of these, but in parallel everything gets found all at once and just stored to the drive. So finding potential off-targets. This is the actual search portion of it now, where you have your CRISPR target of interest and you want to see what your off-target risks are. So potential off-targets are, again, sites with similar guide RNA sequence. I define this by how many mismatches there are between the two sequences. And again, because I've now generated a list of all the targets in the genome, I can just send different sets of targets to different computers and have each computer work in parallel on its own set of targets. So for X number of targets, if I send it to 10 different computers, it would take 1/10 the time it would take to do it linearly. The only thing I need to do is be able to collect the results at the end, which winds up being fairly trivial. And in terms of computational function, I can save the lists as vectors in memory, which basically means that they get piped right into the CPU for analysis very, very quickly. So this is a sample of how sequence comparison would go. Because I save based on the PAM site, I already know the PAM sites are going to be either matched or at least valid for both, so I'm not worried about that. I start after the PAM site at the first base and just keep a tally of mismatches. So it goes and keeps going, and then once it goes over the mismatch tolerance, which in this case was 3, it immediately goes on to the next one. And so I'm just jumping ahead to the matches on this one, which reached the end, only had 3 mismatches, so it's still in the tolerance range. This ends up getting added to the mix of possible off-target sites. One other neat feature of starting at this point is even if I store a longer sequence, which is actually what I recommend for running the program, if I'm running longer or shorter guide sequences for my experiment, I can still search against it so long as the sequence I'm testing is shorter than the sequence I've stored. So sequence comparison, it's highly parallelizable. It requires very limited memory and essentially just 1 core per parallel search. It's able to compare target sites to every potential target site in the genome, and it was taking about 3 minutes using 20 different cores. So acceptable, but I figured there was probably a chance I could do even better because most of the potential sites were rejected within the first couple bases. So I figured I could use this to speed up the searching, and I did that by essentially building a tree structure. And the tree structure, at level 1 of the tree, I have just the first 5 bases following the PAM site. Level 2 of the tree is the next 5 bases. And so now instead of searching every site, I can disqualify over 99% of the sites just based off of which target bin they fall into by their position on the tree. So the results of this, it ended up taking now only about 20 seconds to search on a single CPU, and that's with 3 mismatch tolerance. If the mismatch tolerance is looking for perfect matches, the search time is actually less than 1 second. It requires less than half a gig of RAM, and again, only 1 CPU. And these low resource requirements means I can get resources on cluster systems much more quickly, and I can get much more of them. The increased computational costs were minimal, so it requires an extra step during the indexing, which makes it easy, because you only index once. And it increases the index time by about 1.5 hours, where it was originally about 2 hours, it now takes about 3.5. So what about extending functionality? Because obviously people are not just interested in their own target sites, what if I can guide target selection? So for this, I'll need to think about a ranking scheme for targets. And there's essentially 2 parameters that I think would be minimally required to describe a CRISPR site. Efficiency, which is how often do we hit the target we're aiming at, and specificity, which is how often do we hit something else by mistake. So this is around the time that I actually met a team from Microsoft Research. They were working on applying machine learning to predicting CRISPR target site efficiency, so on-target efficiency. And we decided we'd work together because essentially they're building the ranking scheme that I would eventually need. So use of azimuth, this was developed by Microsoft Research in the Broad, and it's probably the current best system for predicting how likely you are to cut a CRISPR on target based on its sequence. They're right now using a similar approach to develop predictions for an off-target risk. So if you give it a similar match, it'll predict the risk of actually cutting that similar match. And these can be used together to score and rank potential CRISPR targets. And depending on the application, people may want to weight them differently. For somebody trying to do a massive screen in cells, they may want to make sure that they have as much activity as possible because that's really their signal. For somebody trying to do human gene therapy, they're likely to want something that has very exquisite specificity because every potential off-target is a potential cancer-generating event or something equally unpleasant. So extending functionality, this is currently working now, and it's actually been in function for a while, is the user can input an exon or some other stretch of DNA they want to find an optimal target in. The program then breaks it into individual targets, does independent searches on each individual target, and returns a ranked list right now based off of the predicted specificity, but also with information on the predicted on-target efficiency. So with extending functionality, this has been used to design CRISPR targets for multiple experiments conducted at UCLA. This is essentially free for academic use. There's no license required for non-commercial use. It is required for commercial use, however. And it's native to the Hoffman 2 cluster. So anybody trying to run it on campus should have access to the Hoffman cluster and should be able to run it. And then finally, I'm now working on extending this all the way out to where you can either go by gene or by genome, so targeting every transcript for a gene, or targeting even every gene on a chromosome or every gene in the genome. And essentially, this is just going up by level, but I can parallelize at each level to make it reasonably quick. So this is currently under intense development. Building it on the Hoffman 2 cluster, although the big deal right now is eventually porting it over to the Azure system that Microsoft runs to give it a lot more power, because I can pretty much guarantee the Hoffman cluster is not going to be happy with trying to run an entire genome's worth of CRISPR sites. So with that, the message here is CRISPR all the genes with all the CRISPRs. One thing I didn't get into is this system, because I managed to abstract CRISPR sites into just a guide and a PAM, you can essentially index the genome for any guide length and PAM sequence combination and then conduct searches against it. So I've actually run it for Pyogenes CRISPR, I've run it for Pseudomonas CRISPR, and several other species as well. I have several people to acknowledge. First off, the Cone Lab, of which I'm a member. Also the Krakow Lab. Several of those members have given me help with computation, and Pavel has actually even tested some of the CRISPR sites I've developed. The Collaboratory, the Hoffman cluster, where I've run a lot of these tests, Microsoft Research, especially these two individuals who have been working to develop the predictive algorithms, John Dench at the Broad Institute, who did a lot of the molecular portion of developing these predictive algorithms, and then several groups with which I'm associated, the Skeletal Dysplasia Registry, the Orthopedic Hospital Research Center, MCDB, the Collaboratory, and this was funded through both UCLA Collaboratory as well as Microsoft Azure Research grants. And with that, I will take any questions.